And now we will move to the last talk of this session by Dr. Sakuntala Chatterjee from SNBNCBS. Ma'am, you can start presenting. Yes.
Okay, let me share my screen. So the title of my talk is Sensing versus Adaptation in Bacterial Chemotaxis. This work is done in collaboration with Shobhandev Mondal, who is shown in this picture. He is a grad student at SNBNCBS Center, and some of these results are published in these two recent papers. So how a cell is going to behave in an environment is controlled by the reactions which are taking place inside the cell. Now, often such reactions can be noisy in the sense that the protein levels which take part in the reactions or certain other chemicals, they can undergo concentration fluctuations inside the cell, which will definitely affect the outcome. So therefore, many people ask these questions that how this noise, which is anyway present inside the cell, affects the response of the cell, affects the cell behavior. And we answered this question for, this E. coli chemotaxis is the system that we are interested in. And this is about a certain ability of this E. coli bacteria to follow an attractant gradient in the medium. For example, if an E. coli experiences a concentration gradient of an attractant, it has the ability to move towards the region of larger attractant concentration. Similarly, it can move away from chemorepellent. And this motion, guided motion guided by this chemical signal from the environment is called chemotaxis. This motion takes place in run and tumble modes. So during the run, the cell just shows directional motion, and during the tumble, they just undergo random rotation about their axis. Now, when an attractant gradient is present, runs are elongated in the favorable direction and shortened in the opposite direction. For example, in this picture, by using this color, we have represented the attractant gradient. And you see all the runs which have positive component downward, they are extended and the opposite runs are shortened, such that the cell has a net chemotactic drift. Now, how is this possible? So the intracellular reaction network, which makes it possible, makes such behavior possible, has two principal modules. One is called sensing, other is called adaptation. And these two modules are coupled via activity of the chemoreceptors. So these chemoreceptors, which are transmembrane proteins, they can be in two different states, active and inactive. And when this bind to the attractant molecules, ligand molecules present in the cell environment, they switch to inactive states. And this change in conformation of the receptors gets processed by the signaling, the sensing module of the network. And finally, it results in a low tumbling bias, which means when we have large number of attractant molecules present, the tumbling probability is low and the cell can keep running for longer durations of time. So this sensing module processes the signal and modulates the run tumble motility of the cell to propel it in the favorable direction. And the other part of the network, sensing, adaptation, that is responsible for restoring the chemoreceptor activity back to its average value when the activity gets too high or too low. And it does so by changing the methylation level of the receptors using a mechanism of negative feedback. So I don't have time to go into more details about this mechanism. But another important aspect of this signaling network is the cooperativity shown by the receptors. This means that the neighboring receptors, they tend to form tight clusters or signaling teams, and all the receptors which are present in one team, they switch their activity together. As you can imagine, this synchronous switching of activity results in significant amplification of the input signal, which is coming from ligand binding. And this is the reason why E. coli can sense very weak concentration gradient in the medium. Because of this cooperativity, they are very, very sensitive. Now, people are asking the question that what is the effect of noise on this chemotactic performance? So, what is the most important source of noise? It turns out that most important source of noise in this pathway comes from methylation. Why? Because methylation is the slowest possible reaction which is happening in the entire network. Compared to any other reactions which are here, methylation is at least 100 times slower. So therefore, any fluctuation that is present at this level comes as a slow noise, which we cannot integrate out. You cannot get rid of them so easily. And in fact, in an earlier work, we have shown that there is an optimum strength of this methylation noise, which gives rise to the best chemotactic performance. But today's talk, I don't have time to go into this. Rather, I will talk about an experiment recently where people had shown that even in absence of methylation, there is significant noise present in the cell. They have measured the chemoreceptor activity directly, and they had shown that even when methylation module is turned off, chemoreceptor activity shows significant fluctuations. And however, when they disrupt the receptor clustering, the cooperativity of the receptor, when that is broken, then the fluctuations go away. So therefore, it shows that the receptor clustering is an independent and equally important noise source in the pathway. And we are asking the question from there that how this newly found noise source affects the single cell chemotactic performance. So first, let us say, how do we define the performance? You can define it in various ways. One is, for example, how fast the cell is able to climb up the gradient. That is the measure of chemotactic drift velocity. A large drift velocity means a good performance. Or you can ask how strongly the cell is able to localize itself in the nutrient-rich region. Or in a more general sense, a good performance would measure the ability of the cell to distinguish between regions of high and low attractant levels. And we find that there is an optimum size of the receptor cluster for the best chemotactic performance. So this is our results. I don't have time to present the model details, but my model is essentially based on this signaling network. We model the sensing module, adaptation module, and we model the receptor cooperativity. And here we have shown that suppose we consider a linearly varying nutrient concentration in space, then the position distribution of the cell Px also varies linearly, and the slope of that distribution could be one measure of the chemotactic response. And for different values of the signaling team size or cluster size, we find different slope for Px. And when we plot slope as a function of the cluster size, we find there is a particular value of team size for which the slope is maximum. The lower panel shows the data for the chemotactic drift velocity as a function of receptor cluster size. Even here we find a maximum. To measure the ability of the cell to distinguish between higher and lower nutrient levels, we measure the differential behavior when it is going uphill or downhill. So suppose a cell is running uphill, up the concentration gradient, and you measure the average time till its first tumble and compare that with the motion when the cell is going downhill. The difference between these two times is a good measure of performance, because if the difference is large, it means that there is a strong distinction the cell is able to make between these two runs. So this difference shown in this upper panel also reaches a maximum for a specific n. And moreover, this idea can also be used to measure the chemotactic performance for a tethered cell. So what happens for a tethered cell? You have certain flagellar motors. Okay, for all the cells, you have flagellar motors. And when the cell is in the run mode, the motors rotate counterclockwise. When cell is in the tumble mode, the motors rotate clockwise. So people, therefore, often look at a tethered cell and look at its motor response and extrapolate the run tumble response from there in experiments, which is very convenient. That you don't have to track a swimming cell, you just look at a tethered cell. And even for a tethered cell, we can measure this quantity and we find that there is a peak. To understand this behavior, we
you have 1 more minute.
Sorry, 1 more minute. Okay, so I don't have time at all. So so what we have shown here is the activity which controls the tumbling rate. The free energy difference between the active and inactive states has two components, one coming from ligand binding, another coming from methylation. And when the cell is swimming uphill or downhill, when the receptor clusters are small, let's say, then when the cell is the nutrient levels are changing, cell is swimming uphill or downhill, then because of the strong cooperativity, the performance improves, because we have seen that the cooperativity increases the sensitivity and the chemotactic performance becomes better. But when cluster sizes are large, then activity fluctuations increase. And when activity fluctuations increase, then the methylation, actually the adaptation kicks in, it becomes more important. Then the methylation starts playing a more important role. And as a result, this free energy variation is now controlled by methylation rather than ligand density. And the cell is now less sensitive to the ligand environment and responds more to the methylation change. So here we have shown some time series, and the main point I wanted to make here is, for a certain time window, any arbitrary time window, for a large signaling team, we see that this is the time series for change in ligand binding free energy, and this is the change in the methylation free energy. And you see that here the changes are much stronger compared to this. So this is some average quantity we have plotted, the average change in methylation free energy for first few steps of an uphill run. And it is shown very clearly here that as you increase the signaling team size, the methylation, which is these discrete points, they become, initially they were much less than the change in the ligand binding energy shown by the continuous line. And for larger team size, they become comparable, and finally they just overtake this other continuous line. So I won't spend time on conclusions. So we have just shown sensing adaptation competition gives rise to a performance peak. Our results can be tested in experiments. We also propose experiments for tethered cell to directly test this. And these results can also be important beyond E. coli chemotaxis. There are a large number of sensory systems where we have sensing and adaptation modules. And this is a general question people can ask, that whether such interplay gives rise to a performance peak or sensitivity peak for those systems as well. And let me just end this by thanking Deepak and Mustansir. I did my PhD in their group, and I learned a huge amount of academic and non-academic things from both of them. And I won't even try to summarize it in one sentence or so. And also I thank them for allowing us to organize this meeting. We have been trying for the past 10 years. I also thank you all for your attention.
Thank you, ma'am, for such a wonderful talk. We have one question from the BN. Yes. Yeah.
Hi, Sakuntala. So it was just, hello?
Yes,
Are you there? Yeah. So I was just curious that you were finding these peaks more or less at some around some n is equal to 100 some value. So that then signifies something biophysically important, right? A certain size. Do you have anything in mind that what that size may correspond to? And are there examples where like such number, like mathematically it could have been at 50 also or 150, but 100, then is that, does it really occur somewhere? I'm curious.
So one thing that I actually did not get the time to mention is that so this size that physically that corresponds to how large the receptor clusters are. And people can actually measure these clusters in experiment. For example, when they did this experiment on activity fluctuations, they actually disrupted the cluster sizes, and they had shown this activity fluctuations goes down. But there are many things which we have not included in our model, which are actually present in a system. And one particular thing which might affect the quantitative comparison is that we have not considered the membrane curvature energy. To keep our model simple. When the receptors make large clusters, then they also put some curvature energy on the membrane. So that we have not taken into account. So we don't think that this quantitative position of the peak can be verified in experiment directly. However, the main effect that we are describing, and I wanted to emphasize this point, that the main effect that we are describing, this competition between sensing and adaptation, is much more basic than that. Particularly in experiment, people have seen how large teams can generate larger activity fluctuations. So we believe within the same setup, if one directly measures the chemotactic performance like drift velocity, which are highly measurable experimentally, then you can actually see these peaks. But the peak position may not match with exactly what we have.
Understood. Okay.
Okay.
Thank you.
Any more questions? Since there are no other questions, so let's thank Professor Sakuntala Chatterjee.
Thank you.
Thank you, ma'am. And so let's thank all the professors of this session for such a great talks. Yeah, over to you, ma'am.
Okay, thanks Lakshita for chairing the session. Yes, for chairing the session. And Tridib, do we have any other announcement to make?
No, except that we are running a bit late, but still we are meeting at 2:30. We are resuming at
2:30, yes,
as scheduled for the next session.
Yes.
Okay.
Okay.